Okay, in this video we're going to talk about the process of meiosis. Meiosis is really just mitosis 2 times, but the purpose of meiosis is to create sex cells. So it makes sex cells. These sex cells are called gametes, and they are the sperm in the male and the egg in the female. Now, if you remember back to what we talked about in mitosis, you get half your genetics from your mother and half your genetics from your father. In mitosis, we take all of the genetics that make up you double them, and then you split the cells into 2 identical cells. Well, if we were to do this with sex cells, then each sperm would have 46 chromosomes, and each egg would have 46 chromosomes. And that's way too many chromosomes. We know that each cell has to have only one set of 46 chromosomes. So, here comes the process of meiosis. In the process of meiosis, it actually splits the amount of chromosomes that we have in half, so that each sperm and egg has 23 chromosomes in it. So, in a regular somatic cell, goes through mitosis, and they have 46 chromosomes. Sex cells, or gametes, have 23 chromosomes each. That way, when they come together, you end up with a full set. You get half from mom and half from dad. So somatic cells are called diploid. It means 2 sets of chromosomes. Gametes are haploid, which just means 1 set of chromosomes. That way, when you get your chromosomes from mom and from dad, they come together and you end up with 46 in a fertilized zygote. Alright, so the process that we go by for making these sex cells is called meiosis. And really, like I said, it is simply mitosis 2 times. The first time, we're going to split up what are called homologous chromosomes, and I'll go into that in just a minute. The second phase of meiosis, or mitosis 2, is where you split up those sister chromatids, and it looks the same as regular mitosis in a regular somatic cell. Alright, so for this, you can see that I've fussed up my paper a little bit, and I also have 3 colors. So I'm going to use these 2 colors here to represent my chromosomes that I draw, because in meiosis, we create unique cells, unique pairings of chromosomes. I'm going to use these colors to identify the different chromosomes. So take a minute, pause the video, mark up your paper, get some colors so that you can draw this along with me. And you can see also that I've drawn the nucleus disappearing, I've drawn my centrioles, and I've labeled, sorry about metaphase, it's a little messy, but I've labeled prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. And down here, I've drawn duplicates of those, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis again. Alright, so let's get started. For the purpose of this video, I'm simplifying our chromosomes, and I'm only using 4 chromosomes total. If I were to draw more, or 2 actually homologous pairs, if I were to do more than that, it would be too messy. So I'm going to use green, and I'm going to make a big X and a little x. And I'm going to use red, and I'm going to make a big X and a little x. Alright, so there's this unique process that creates even more genetic diversity. It's called crossing over. So what happens when these 2 chromosomes, these homologous chromosomes, let's label those first. These right here are homologous chromosomes. Homologous chromosomes, remember from your word parts, homo means same. So these are similar chromosomes, but they are not identical. What they do is they have genetics from mom and genetics from dad. So they'll code for the same genes, but maybe not the same trait. For example, hair color. Both of these chromosomes might carry the genetics necessary for hair color, but one of them might carry black hair versus blonde hair or brown hair. So that's where they're similar, but not identical. And a really nice thing happens when you have these homologous chromosomes that get really close together is on occasion something called crossing over can happen. And what that means is that a little piece of this green one comes over and switches places with a spot on the red one. So now we have 4 completely unique chromosomes. So this process is called crossing over. And it's where we get genetic diversity. And it is right here. How we get a little bit of one chromosome switching sides to the other one. And this is how that even though you get your parents' genetics, you don't look identical to either of your parents because they can mix together. All right, so now we've got our everything that's happening in prophase and mitosis is happening here. The nucleus is disappearing. Those centrioles are moving to the poles of the cell. The chromatin is wrapping itself up into these nice chromosomes, and these homologous chromosomes are finding their pair and moving next to each other. So in metaphase and mitosis, the chromosomes line up single file all the way down the cell. In meiosis 1, we are splitting these homologous chromosomes. So they're going to line up in their pairs next to each other. And those spindle fibers are still going to attach to those centrioles. They're getting ready to be pulled to the opposite poles of the cell. We have them now being pulled apart to their poles. Still with those spindle fibers pulling those chromosomes to either side. Now in telophase, we have our nucleus starting to reappear. We have our chromosomes in their individual, or starting to be individual, cells. So now instead of being genetically identical, these two cells over here when we get to cytokinesis are going to be unique. So what we have done here is we have halved the number of chromosomes. We started here with 4 X's, and now we have 2 in each one. So in meiosis 1, I'll write it in red. We are splitting homologous chromosomes going from diploid to haploid. Because remember, each of these, over here before crossing over occurred, these are sister chromatids. So this one was identical to this one. They'd already been duplicated during interphase. Now that we had the crossing over occur, they're not identical anymore. But these ones down here, these small ones, stayed identical the whole time. But now what we've done is we've taken the number of chromosomes and cut it in half. So now what happens in meiosis 2 is we are going to split the sister chromatids. So the way we do that is we're going to transfer, we'll move this up a little bit, what we have here, and we're just going to split them up like regular mitosis. We've already got our duplicated chromosomes, so we have a brief interphase, but there's no duplicating of the DNA that's already been done. So now we come down here, and I'll put my green ones on the top, and I'll put my red ones on the bottom. So they're going to go through, again, we have our nucleus disappearing, we have our centrioles reforming, and in metaphase this time, it's going to look just like mitosis. Got our big one and our little one. Those spindle fibers are lining up in the middle of the cell. In anaphase, now we're going to pull those sister chromatids apart. Still those spindle fibers doing their job, pulling those sister chromatids apart. Now in telophase, we now have a tall one and a short one. A tall one and a short one. Tall and short. And tall and short. And our nucleus is starting to reappear. We have taken our chromosomes and split that duplicated chromosome. And now in our individual cells, we end up with 4 unique haploid cells. So these 4 cells become your 1 genetically diverse 2 egg and sperm.